Hello, welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, this is lecture number 21, where we will start indole alkaloids. Early step of biosynthesis will be covered in this lecture, and then slowly we will move into the late stages and the advanced aspects of it. So, the concepts to be covered in these lectures are indole alkaloid basic structure, examples of diverse indole alkaloids, then early steps of indole alkaloid biosynthesis in general, then specific alkaloid pathways in Cinchona, Rauwolfia and Catharanthus, and then if time permits we will start discussing hairy root as a system for indole alkaloid production. Let us go to the board now. So, the indole alkaloid the basic structure is like this. Here there are two things. This is the pyrrole ring and this is the benzene ring. So, these two rings basically fuses and form indole alkaloids. The precursor amino acid of indole alkaloid is tryptophan. Tryptophan PHAN. Okay. So, what happens that these indole alkaloids also we use the term terpenoid indole alkaloids. This means a terpenoid moiety will also join with the indole ring. So, now before we come to that, let me give you some examples of indole alkaloids, and also I will write the first intermediate from where different alkaloids accumulate. So, the major important intermediate, which is a very active compound, which is called strictocidin. So, from strictocidin different alkaloid pathways emerges. For example, the well known vinblastine and vincristine. These are called dimeric indole alkaloids. Dimeric means that two monomers are there. So, what are these monomers? These monomers are vindoline and catharanthine. So, in other words, vindoline and catharanthine, they are monomeric alkaloids, and these two joins together and form dimeric alkaloid. Okay. Now, apart from vincristine, vinblastine, vindoline and catharanthine, there are other examples. For example, okay, let me put the arrow first. Vincamin. So, vincamin is found in the plant called Vinca minor. whereas all these alkaloids what I have written in the right side, they are all found in the plant called Catharanthus roseus. English name is periwinkle. Okay. Now, apart from this, there is another alkaloid which will also produced by Catharanthus roseus, which is also monomeric, which is called azmalicin. Okay. Now, azmalicin is produced by both Catharanthus roseus as well as another plant called Rauwolfia serpentina. So, its common name is Sarpagandha. So, apart from azmalin, there are other indole alkaloids such as azmalin, serpentin, raucafrin, raucafricin. Okay, raucafricin. These are also produced by Rauwolfia serpentina. Now, let us see another example which we will write here, the quinine. Quinine alkaloids formed from the indole alkaloids, and a good example of quinine all of you know, this is cinchona, CINCHONA officinalis. Now, the latest member in this group which I will write is strychnine. So, strychnine is produced by the plant called Strychnos nux vomica. So, nux vomica is a well known homeopathic medicine. So, all of you have heard about it, but strychnine is very poisonous. So, these are the different indole alkaloids produced from the basic skeleton, which is strictocidin. Okay. What we are going to cover in this class? We are going to cover, not in this class as such, in the in this class today we are going to cover these aspects. We are going to cover these aspects, and see how much time permits, so that we can see these steps or not. But eventually we will also cover all these, that means the details of this vincristine vinblastine formation from vindoline and catharanthine, and I will take a separate class on strychnine, because strychnine biosynthetic pathway has recently been discovered using molecular tools. So, that we will also cover in one of the subsequent classes. Now, with this, let us now move into the basic biosynthetic pathway. Here now I will use the term terpenoid. Early steps of terpenoid indole alkaloid biosynthesis. Pyruvate joins with glyceraldehyde 3 phosphate and it makes eventually isopentenyl pyrophosphate. So, these details we will see when we will start discussing the terpenoid plus pathways in another week. Okay. And this ultimately leads to the formation of geraniol. So, geraniol and from geraniol it makes loganin and from loganin it makes secologanin. So, and other route originates from Shikimate pathway. So, therefore, how we will write? We will write in this way Corismate CHO which makes anthranilate and that makes tryptophan. Now, tryptophan is converted into tryptamine okay, I use this side. And this secologanin and tryptamine joins together and forms the strictocidin. So, this is an important step. The enzyme which is responsible for this joining is called strictocidin synthase or STR. So, STR stands for strictocidin synthase. Tryptophan to tryptamine is converted by an enzyme called TDC. TDC means tryptophan decarboxylase. Loganin to secologanin is formed by secologanin synthase SLS. Okay. So, now strictocidin is basically a molecule containing a sugar or glucose. So, strictocidin I will put it in this side. Okay. by the action of an enzyme called beta glucosidase, converted into strictosidine aglycone. And this is basically a very active molecule, because this subsequently converted into different products. So, I am going to show you the different products formed from strictosidine aglycone. So, strictosidine can make three different components. Strictosidine aglycone, one is the cathenamine. Sorry, CATHENAMINE. Another one is 421 dehydrogaseosizine. Dehydrogaseosizine, and it can also converted to epicathenamine. So, let us put the arrow. Now, 421 dehydrogaseosizine is very important molecule, because this subsequently moves towards the formation of ultimately the dimeric indole alkaloids. Ultimately, now there are cathenamine is also an active molecule, because that leads to the formation of other indole alkaloids of the pathway. So, let me show that in another slide. Strictosidine and then what I said, that it forms either 421 dehydro sorry. Okay, and the other one is that it can also form 4,5 dehydro whatever may be. So, this may be the plant specific. Now, we will see the left side. So, that means strictosidine now makes this 4,5 dehydrogaseosizine, and this will be converted into, I just use the board. So, I just put it in arrow this way, converted into a compound called polynuridine aldehyde. This is also denoted as PNA. These PNA subsequently converted into 16 AP velocimine, and that subsequently converted into diacetyl vinodine, and diacetyl vinodine will be converted into vinodine, and vinodine subsequently converted into bromelain, okay. And so, let us put the arrow. So, diacetyl, from diacetyl vinodine, there is an enzyme called vinodine synthase. Oh no, so it's here, yeah. Which converts diacetyl vinodine to vinodine, and in this process it requires a molecule of acetyl CoA. Okay. Vinodine to valproate is basically a hydroxylase, and now bromelain will can convert into a molecule called raucafricin. So, raucafricin. So, this is basically raucafricin, and it is stored there, and then this can be also get back to valproate. So, there is one enzyme called glucosidase that basically converts back to raucafricin to bromelain. So, this is a glucosidase here. And the other one should be a glucosyl transferase, and then I will talk about little bit of raucafricin, but let us see the where this pathway ends. This pathway moves further and makes 1,2 dihydro bromelain, and this in the next steps form acetyl noradrenaline, and this converted to noradrenaline, I will take a bit more space. And noradrenaline finally converted into adrenaline. So, this last step requires a methyl transferase. And the other step which requires reductase. And acetyl to no, sorry, this is not this, reductase will come here. And this will come acetyl esterase. So, ajmalicin can be converted into ajmalin can be converted into serpentine by another reaction. So, serpentine. Now, in cell cultures of Rauwolfia serpentina, sorry, RAUWL, sorry. This pathway has been elucidated. Okay, ajmalicin, serpentine, these are important molecules, particularly for reducing the hypertension. Okay. And other important point which I would like to tell, that when the normally in nature this compound which is raucafrin, raucafricin is only found in Rauwolfia capra. CAFRA. However, when Rauwolfia serpentina cell culture were established, and with appropriate media combination, Rauwolfia serpentina were able to produce raucafricin at the level of 1.2 gram per liter, and ajmalin at the level of 0.3 gram per liter. This is indeed very interesting, because normal wild type Rauwolfia serpentina, this alkaloid remain undetected, whereas in the cell culture with media manipulation, rauc capricin can be detected at a higher amount. So, that means the cell culture of Rauwolfia serpentina can be used as a system to produce rauc capricin. That is the interesting aspect of it. So, vinodine, what I have said, so vinodine is basically the acetylated indolanine alkaloids. Okay. Now, one point here is this, that means that why this is happening, in Rauwolfia serpentina. So, that means what happened, that in Rauwolfia serpentina cell culture, the pathway is getting somewhat deregulated. There lies the question. So, this question needs to be answered. So, with this I stop this class here, and in the next class we will look into further details. So, thank you very much.